Hey, this video will show you some hacks that you can follow when you are completing scientific method questions. So recall that we do have some task verbs: identify, describe, calculate, explain, justify, make a claim, propose a solution. Today, we're going to be discussing designing a controlled experiment. So when you are following this task verb, what you should be doing is following the scientific method. You will be asked to establish some of the variables, and you may be prompted to predict some results. There is a cheat sheet, like worksheet, that I have, and you very likely have seen it, but this is my explanation of the entire process. So these are the steps that I'm going to be following. Here is our prompt. Temperature and respiration rate are intricately linked in living organisms. As temperature rises, molecules within cells gain kinetic energy, leading to an increase in metabolic processes, including respiration. Higher temperatures generally boost the rate of respiration in organisms, as enzymes and biochemical reactions function more efficiently. Conversely, cooler temperatures tend to slow down metabolic processes, reducing respiration rates. This connection is crucial for understanding how temperature fluctuations impact the survival of organisms across various ecosystems and serves as a fundamental principle in ecology and physiology studies. A group of students plan to demonstrate this in a controlled experiment using yeast, a single-celled eukaryotic fungus. So the first step that we want to follow is we want to ask a testable question. And this is the only time that you should be asking a question. And you can format it in this, or structure it in this format. So what is the effect of blank on blank? So what is the effect of the cause on the effect? So what we're going to be doing is we want to compare some of the factors that were described in this prompt. And the cause, as we learned, was temperature. Now, temperature is related to the speed of these metabolic processes, and the metabolic process that was mentioned in here was respiration. So the rate of respiration. So what is the effect of temperature on respiration rate? You'll notice that I have color-coded this. So this is red, this is blue. This is going to serve as a staple throughout this entire activity. So follow along with the colors. Now what we need to do is write our hypothesis. And I know a lot in middle school you may have learned if, then, because. You don't have to write because that will be explained throughout the rest of the controlled experiment. Now, the if, notice it is red, and the then, notice it is blue. So the if will be something related to temperature, and the then will be related to respiration rate. Now, the part that says if, we're going to be adjusting. The part that says then, we're going to be measuring. So if temperature, then respiration rate, that doesn't make sense. So we want to make it into a sentence. So if you raise the temperature, then respiration will increase. Now that was directly from the written prompt. You could also say if you lower the temperature, then respiration will decrease. Now, conversely, you can also have, a wrong hypothesis. If you raise the temperature, respiration will decrease. That is also acceptable because it will be proven correct or incorrect throughout the experiment. But for the purposes of this video, I am following this hypothesis. Now what we want to do is we want to set up our experiment. So what we need to do first is we need to identify the variables. The independent variable is the first part. Independent, here's what I always remember, starts with the I, independent if. And that involves two different groups. We have a control group and an experimental group. So our independent variable, again, is literally just the part that's in red. And inside that independent variable, we have the control group and the experimental group. The control group involves either no change, nothing additional has been added, or it is under normal conditions. And the experimental group is where we are adjusting just the independent variable. So in this case, we are only adjusting the temperature. Everything else will be kept the same. So if you look, we're going to have, different setups. These will be test tubes with some sort of solution. There's going to be yeast inside, and all the conditions are the same, except this one will be at a different temperature. I chose for this example room temperature. There's many different controls you could use. You could also have it be 37 degrees Celsius. That's normal body temperature. I would say that don't choose 0 degrees Celsius, because that is the freezing point of water, so that really doesn't make sense. But when you're having temperature, I would say either room temperature or normal body temperature would probably be a good idea. Now in the experimental groups, I want to adjust this. So let's go 25, 30, 35, 40, 45 degrees. Any higher than that, you're going to have some damage to these yeast, the yeast fungus. But you could go up to, 100 degrees, the boiling point of water. That is also acceptable. Now, the dependent variable is the other part. This is the part that we're going to be measuring. The independent variable basically is going to dictate the changes in the dependent variable. So as I adjust the temperature, something will happen or should happen to the respiration rate. So I'm going to be measuring the respiration rate. Now, the next step is we are going to be collecting our data. So the first thing we want to do is construct a table. When you construct a data collection table, you're going to have your independent variable on the left, and your dependent variable is going to be on the right. Now, the independent variable, you do not have to indicate which one is the control. If you do see a data table, they may ask you which one was the control, and you should be able to determine which one had either nothing additional added or had normal conditions. Now what we're going to do is we are actually going to collect this data. So in this situation, you would be conducting this experiment. I have populated this chart with some numbers. So if you look, here are the numbers, the respiration rates, and this indicates the number of gas bubbles that were counted in a minute. So you do notice that there tends to be a general increase, and then it kind of levels off and maybe decreases a little bit. So once you have your data collected, you will need to construct a graph. And when you construct a graph, obviously you're going to set up a grid with your X and Y axis. The independent variable goes on the x-axis, and the dependent variable goes on the y-axis. And then what you need to do is set up a scale. And with your scale, I don't have the scale indicated here, just in the interest of space, but you really want to count by a standard number, like 5s, 10s, 20s, 1, 2, 5. Don't simply just copy the numbers down that you see in this data table. So again, you want to count by the same number every single time. You do not have to start with 0. This point right here, the origin, does not have to be 0, 0. It could be any number you're starting at with the temperature. Like for this one, I could start at 20 degrees. For the rate of respiration, I could start at 10. I could also start at 0 if it fits and if I am not adding additional boxes at the end. Now, once I graph the data, it will look something like this. Always we want to make sure we include a title because we may be prompted to insert a title. The last step, what we need to do now is we need to describe the relationship. So what is the relationship between the independent and the dependent variable? So you can follow this format. As the x-axis increases, again, as it increases, you're going to the right. What's happening to the y-axis? Is it either increasing, decreasing, or remaining the same? So as temperature increases, the respiration rate also increases. This was a very simple experiment. I hope that this video was informative. I thank you for your attention, and I will see you soon.